Hi everyone, and thank you for being here. I am Karin Gemini, a research fellow from the University of Würzburg in Germany, and I am your host for today's webinar. So this webinar series is a collaborative initiative between the Society of Neuroscientists of Africa, the African Academy of Neurology, Trend in Africa, and other organizations. So with this webinar series, we aim to bring experts in neuroscience from all over the world and students and university staff, especially in Africa, together. So this provides a great learning and exchange platform and helps also our neuroscience community to grow. So this is a great opportunity to get guidance and mentorship from experts. And today, it's really my pleasure to welcome an African neuroscientist here as a speaker. I am very excited to host Dr. Arif Ahmed, a rising star of neuroscience, if I can say it that way. So Dr. Ahmed is a young fellow with already an impressive work only a few years after his PhD. He is currently a Hannah Gray Fellow at Howard Youth Medical Institute at the university, at the Brown University in Rhode Island in the US. For those who don't know, the prestigious Anna Gray Fellow Program, it's a program that funded a postdoctoral fellow, and Dr. Hamid has been funded with an amount of $1.4 million for the next 8 years to support his research. Uh, what can I say about Dr. Ahmed? Dr. Ahmed finished his high school in Addis Ababa in Ethiopia before moving to the US, where he completed a Bachelor of Science in Neuroscience at the University of Minnesota before doing his PhD in neuroscience at the University of Michigan. He's interested in brain computation for flexible behavioral control and learning with a special focus on the neurotransmitter dopamine. So I am sure he's going to touch on that in his talk today. His work combines sophisticated rodent behavioral approaches together with a variety of methods for neural activity AC, including high-density electrophysiology and two-photon microscopy. And his work has been published in prestigious journals like Nature Neuroscience. And with this groundbreaking research, also, he received several major coverage. I just Googled a little bit and found that he was very recently nominated on the list of inspiring Black scientists in America. So press release, February 2020. So we are really happy to have you here, Dr. Hamid. You're very welcome. And I want to remind the audience that the questions can be posted in the chat section at any time during the talk, and Dr. Hamid will answer them during the Q&A session. Now, I will leave the stage to you, and we are eager to hear what you've prepared for us today.
Awesome. Thank you so much, Karina. That was a very nice introduction. I'm also quite excited to talk, to the African community of neuroscientists to share some of the work that I've been doing, both as a graduate student and also some of the work that I've been working on as part of my postdoctoral training. So generally, I'm interested in how the brain orchestrates flexible and adaptive behaviors. And a really important challenge that it needs to meet is kind of the demands of the present and also the future, yeah? And what I mean by this is that, the present requires that we use previous learned experiences so we can decisively act in the moment right now, whereas the future requires that we store or learn kind of from past experiences so that in the future we'd be very excellent agent at trying to retrieve rewards from the environment, for example. We know quite a bit that a network of brain areas that includes the cortico-basal ganglia circuitry and the neurotransmitter dopamine really help to resolve these challenges. And my long-standing research goals have really been to understand how the neurotransmitter dopamine affects this basal ganglia circuitry to provide quantitative descriptions about the decision signals being relayed by dopamine, and also what biological substrates or circuit specializations help to implement or support kind of these specific computations that I'll elaborate much more on as the talk goes on. And I try to go about doing this by designing very specific tasks that help us understand how different behavioral demands end up leveraging these circuit specializations and computational specializations of the circuit to really afford adaptive and flexible behaviors. Now, I hope I don't have to, convince you too much that dopamine is super, super important neurotransmitter in the brain. It has, important roles in several normal functions that include reward learning and decision making, but also in coordinating and invigorating or motivating various locomotor outputs from animals and humans, but also in the more cognitive domain. It's quite critical for organizing executive functions and regulating working memory and other things that we can call broadly classify into kind of cognitive effort and being able to stay focused on task and so forth. And obviously, when dopamine systems and the underlying circuitry gets perturbed, these normal functions manifest themselves as various neurological and psychiatric disorders in the appropriate domain of dopamine's role. And so, as many of you may already know, a variety, most all of these disorders, the therapeutic direction is really to provide various forms of supplementing or antagonizing various dopamine receptors. And so understanding how dopamine affects the circuitry ends up being quite important in expanding and understanding the involvement of dopamine in these various disorders as well. So this is a side view of the rodent brain, and dopamine cells tend to live here in the midbrain. And there's relatively very few of them, but what's quite impressive about their anatomy is they make these really dense axonal projections along the medial forebrain bundle to arborize out here in the forward part of the brain, the forebrain. While they do go to the cortex, they quite intensely arborize within the striatum, this input nucleus to the basal ganglia. And if you look at studies that have reconstructed individual dopamine cells, it's actually quite awe-striking because here's a dopamine cell that lives in the SNC and it's making its way towards the forward part of the brain as it arborizes across very large territories of the striatum here. And here are additional examples, a single cell occupying very large territories of the striatum. Now, in terms of what does dopamine do? Dopamine tends to regulate this underlying cortical basal ganglia gate that itself has the role of deciding what is worth doing, to select among candidate options or proposals from the cortex, and acting as a gate to either accept or reject these, and through a thalamic relay, send these back to the cortical subregions to refine these proposals. And the dopaminergic input or the modulation by dopamine really allows this network to be plastic to gate based on what has worked before. And this is really, the consensus is really, this is the brain locus for reinforcement learning. A link to some literature in the computer science also, which I'll try to make in just a little bit. At the cellular level, we know that dopamine accomplishes these two roles, either by affecting the plasticity of this cortico-basal ganglia input by allowing these dendritic spines here, for example, if you cause a coincidence of glutamatergic input and dopaminergic input together with depolarization of these medium spiny cells, you can cause these spines to grow here in real time. And so this is a kind of a structural piece of evidence that suggests dopamine can facilitate plasticity by regulating the potentiation of the synapse, but it also has the capacity to change the excitability of these medium spiny cells on much shorter timescales of milliseconds and seconds. At a more behavioral level, in terms of what dopamine does, we got very early insight into the role of dopamine, mostly from kind of post-mortem analysis of Parkinsonian patients wherein dopamine cells tend to die off. And because the key deficit in Parkinson's patients is the capacity to initiate, invigorate, and coordinate various locomotor outputs, we had early evidence that dopamine must be important for movement. Now, these views have been refined over the years into one that suggests that dopamine is actually important for motivational arousal, whereby it energizes actions towards desired goals. And this is really at the nexus of transforming whatever is valuable from the environment into movement to go pursue those goals. A parallel literature also suggests that dopamine is very important for learning and reinforcement type functions. These are some seminal set of studies by Olds and Milner, wherein they implanted the animal, the rodent, in different parts of its brain and checked to see which subregions the animal is willing to press a lever to get intracranial brain stimulation. And they found that these cells are actually clustered right around the midbrain dopamine cells, but also included other areas like the hypothalamus. Much later, we got more direct electrophysiological evidence that dopamine activity patterns were actually involved in reinforcement learning. And here I'll just summarize some results from one of the earliest and most famous results from Wolfram Schultz and colleagues, wherein if you squirt a little bit of juice into the monkey's mouth and are recording dopamine directly from dopamine cells, you find that dopamine cells tend to burst, suggesting that they're pleasantly surprised by this juice that has come out of nowhere. But if you pair a cue, a tone, to the reward multiple times, what happens with the dopamine cells is they now respond to the onset of the cue itself and are no longer by delivery of this reward, suggesting that these dopamine cells may be helping the animal learn the relationship between this cue and this reward itself. Now, this and many other studies across the decades have been kind of formulated into a reinforcement learning computational description of dopamine that suggests that dopamine is important for learning functions, that it signals this reward prediction error terms, effectively the first derivative of a value gradient as animal tries to learn which states are valuable in the world and or which of its actions were so valuable in terms of producing rewards in the world. So just to summarize what I've told you, as an agent interacts with the world through its actions, the environment provides these reward feedback. And so this dopamine motivation hypothesis really tries to emphasize this performance role of dopamine, online role, whereas these dopamine learning hypotheses suggest that it's really important about incorporating this reward feedback so that future behaviors can be appropriately targeted. Now, I should be the first to tell you that both of these hypotheses are indeed true. Dopamine does have this dual role, but a really important challenge in the field for a long time has been reconciling the simultaneous role of dopamine both for learning and performance functions. So these are what I went after, is to resolve this rift in the theoretical account for dopamine in graduate school to ask, how does dopamine really regulate both learning and performance simultaneously? And I did this in rats doing this trial and behavior task, where they were provided with an option of going either right or left that are themselves probabilistically rewarded. And what these animals tend to do across learning is, if, for example, the left-hand arm is valued at 90% reward and the right-hand arm is valued at 10%, these animals tended to increase their probability of choosing the left-hand arm as the block progressively gets completed. Whereas if the probabilities were reversed and the left-hand arm is not as good as the right-hand arm, these animals tended to avoid the left-hand choice, suggesting that they can adaptively learn and express these adaptive choice behaviors. These animals also get quite good at expressing adaptive vigor. So if, for example, 10 out of the last 10 trials were rewarded, the latency to start playing a single trial, initiate a single trial, becomes very, very short, and these animals knock out these trials back to back to back. Whereas if animals if the game is not yielding as many rewards as the animal likes, the animal is quite lousy to start initiating to play the game, suggesting that they're expressing these learned values through some form of adaptive figure. And so I went on to do direct dopamine measurements within the ventral striatum as animals are performing this task, as you see here in this video scrolling, together with optogenetic manipulations of dopamine activity within the midbrain dopamine cell bodies, together with a very nice reinforcement learning model that captures the ongoing decision process as the animal is making these behaviors, to effectively argue that dopamine release within the striatum multiplexes these motivational and learning signals. And we made the case that this is accomplished by conveying what we called a reward expectation or value signal. And this is actually quite a simple concept, and I'll just describe it here in this slide. So these reward expectation or value signals are basically temporal discounts of future anticipated rewards. Okay, they just tell you that as time moves along, or you physically get closer and closer to reward, they tell you that there's a gradient as you're getting closer and closer to reward. Because these value functions scale appropriately, if rewards were less probable or more distant in time, the levels of these discounted value functions are actually really, really useful as a motivational signal as to when an animal needs to decide whether or not it's going to pursue these distant goals. It can use these levels, absolute levels of this value signal. Now, unexpected reward cues that tell you reward is now going to be delivered will rapidly adjust these reward expectation signals. For example, here, an initial pessimistic expectation upon knowing that you're going to get a reward will correct this value trajectory to the discounted value of the now certain reward. Whereas an optimistic expectation will be corrected to a lesser extent. And because prediction errors are by definition rapid deviation from unexpected deviations from value, these corrections or deflections are themselves reward prediction error signals. And so the key component of what we argued is really to suggest that if you continuously and dynamically encode a value signal, you get both motivational signals and reward prediction error signals for free that can regulate both ongoing behaviors and can induce plasticity to regulate future behaviors. And this is exactly what we demonstrated in the experimental data, wherein the dopamine release across multiple timescales is highly correlated and co-varies with model estimates of value signals, and that rapid deflections in these dopamine levels encode reward prediction error signals. We also causally showed that dopamine in the brain can be used to regulate immediate motivation because we can excite dopamine cells to cause the animal to play the game more sooner, shift the latency distribution. But also if we inhibit dopamine cells, we can shift it in the opposite direction. And we can also drive learning because, again, if we excite dopamine cells, these animals will be more likely to repeat the choice and less likely to repeat the choice if we inhibit dopamine cells. So just to briefly summarize what I've told you guys thus far, that within the nucleus accumbens motivational hotspot of the ventral striatum, dopamine there tends to encode the value of motivational engagement or work. And we thought that this is a really neat coding or multiplexing strategy that dopamine utilizes to shape both ongoing behaviors and future behaviors. Now, in a follow-up with a postdoc in the laboratory, we also recorded the activity pattern of midbrain dopamine cells and were surprised to find that they were faithfully encoding reward prediction error signals, suggesting that there may be disjoint coding of dopamine release and spiking in terms of what decision signals they care about. You know, these results, these findings were initially surprising, but we thought they may actually be implementing an adaptive circuit strategy that may allow target subregions of dopamine receptive areas to tailor dopamine according to their own computational requirements. Now, the problem is it remains unknown whether or not brain-wide dopamine levels are systematically dissociated or decorrelated according to some specific computational or circuit mechanisms. Now, there's a lot of clues to suspect there should be, because if you look at these recipient dopamine subregions, they tend to be organized into these repeating motifs that span the motor, cognitive, and limbic domains of corticobasic ganglia function. We can also distinguish these different parts of the underlying striatal regions based on which complement of glutamatergic cortical input they get into different parts of the striatum, or also by the complement of molecular phenotype of receptors that they express, which include calcium binding proteins and various other things. You can effectively cluster out different parts of the striatum. And more importantly, they're also organized hierarchically into these spirals that, for example, Susan Haber described a long time ago, that is itself an increasing level of abstraction or time scale of planning. Across these different parts of the striatum, dopamine still needs to be able to perform its dual roles that I mentioned earlier. That is, it should allow that circuit to energize or express the learned values and still affect plasticity to affect how that subregion stores or learn new values based on environmental statistics. Now, a detailed description of how brain-wide or dopamine release is heterogeneous is really a significant gap in our understanding at the moment. And this is what I set out to tackle during my postdoctoral work, to really reevaluate the current notions or frameworks of functional specialization of dopamine across really large swaths of the striatum. And I just want to give you a premiere into the current framework suggests that midbrain dopamine cells are highly correlated, and that they have this really impressive divergent anatomy that I showed you guys in earlier on. And a combination of these suggests that a reward prediction error signal that's being encoded here is really being broadcast to all striatal subregions. Whereas the problem with this is that it's really not computationally advantageous, because you tend to run into a structural credit assignment problem, wherein if different parts of the corticobasic ganglia circuitry are involved in gating through a decision to different extents, this is indicated by the size of these circles, just blasting everybody with a scalar or global reward prediction error signal is really impoverished. And a suggestion in the field had been that you vector weight these reward prediction error signals that arrive at different parts of the striatum. And so these dopamine signals would be tailored to the computation of this underlying subregion itself. Now, this has been shown to improve learning in some complex tasks and simulations, and there is some evidence that there's some plausible anatomy, but it still has not been demonstrated directly. Now, what I am suggesting, or this revised view that I'm suggesting, is that there must be within the striatum or at the target location, some form of specialized dopamine signals that end up tailoring dopamine to the underlying subregion's own computational requirement. And so the key challenges for advancing this revised view is that there must be some fundamental organization of the activity pattern of dopamine within the striatum itself, while still remaining true to the circuit general computational description of dopamine that involves both learning and performance functions. Now, today, I hope to suggest to you that these dopamine waves are these fundamental organizational rules, and they tend to vectorize or codify reward prediction error and value signals across different parts of the striatum. So to experimentally get at this question, what I set out to do was to image the really large scale organization of dopamine axons within the striatum. And the approach that I took was to get a mouse line, a DAT-Cree mouse line that labels dopamine neurons and inject at a Cree-dependent GCAMP6, a fluorescent indicator for calcium levels within these cells. And injecting there will cause an expression of GCAMP not only within the cell bodies, but also across the axonal arbors. And this will allow me to get an assay of how active these axons are. I also used an alternative approach wherein I directly inject a very new fluorescent indicator of dopamine that has the capacity to bind dopamine and fluoresce to give you a signature of how dopamine may be changing within local striatal subregions. Once I make those injections, I have this optical preparation where I remove the overlying cortex to get optical access to this dorsal surface of the dorsal striatum. And this preparation is big enough so that I can assay 60 to 80% of the dorsal surface of the dorsal striatum. And this is what it looks like right after surgery. But once it heals up and the blood clears up, you can see I get really nice access. This is the lateral ventricle right here. I get really nice access to the dorsomedial striatum, to the more lateral parts, the dorsolateral striatum, and also some of the tail portions of the striatum. And I put these animals on a head fix preparation for both 1 photon and 2 photon microscopy. Now, this project that I'm about to describe is really not short of detail, and I will, for reasons of streamlining the talk, I will only talk about a subset of the results. But for those of you who are interested in reading more, please check out this preprint that we have up on bioRxiv, and hopefully it will come out soon anyway. Okay, so just for those of you who may not be too familiar with the anatomy of the dopamine system, I just want to give you a flavor of what the field of view looks like. So here is the midbrain dopamine areas, and these green areas are the striatal structures. And dopamine cells make these dense axonal arbors and really arborize out into the different striatal subregions. And so the field of view I'll be telling you guys about is a top-down field of view that looks at activation of dopamine axons here at the dorsal surface of the dorsal striatum. Okay, so the very first and super exciting result that we found is actually that dopamine axon activation is organized into these spatially and temporally smooth and continuous activation patterns that I'll call dopamine waves. And this is observed in the calcium activity of the axons underlying this subregion. And I just want to give you a sense of scale. This is 1 millimeter in a mouse brain, and this field of view is 3 millimeters. So these are waves that are traversing very large territories of the striatum. This pattern of activation is also observed in the concentration of dopamine. This is using that sensor that I told you guys about, which binds dopamine and fluoresces. So these really complicated spatiotemporal patterns are observed both in the activation of the dopamine axons and also dopamine release. I just also thought I'd give you guys a sense of what it looks like with animal behavior itself. Here's an animal which is running, you get spatiotemporal activation patterns, but also when the animal stops, you get these patterns as the animal is still not doing anything and not experiencing any sensory signals. Now, the really cool part is these waves tend to coordinate dopamine input to functionally related areas. And what I mean by this is if you look at the activation profile of dopamine in small territories of the striatum, you can see that different parts of the striatum experience different patterns of dopamine input. And if you look at how well these signals are correlated, depending on how far away these striatal subregions are from one another, you find that nearby areas have a very highly correlated dopamine input, but the further you go for the two pairs, the more decorrelated these areas tend to be. And this is true both for GCAMP and the D-light signal. But what we thought was quite interesting was that this was true mostly for these triangles here, which is for medial lateral distance of the two subregions, and not for anterior posterior distances of these paired correlations. And that suggested that dopamine may be systematically organized on the medial lateral axis. And so to identify this a little bit better, we used standard cluster methods to identify subregions that may be receiving hierarchically related dopamine inputs. And what we immediately find at the cluster limit of 2 is the dorsomedial striatum, what we know from functional studies to be the dorsomedial striatum and the dorsolateral striatum immediately fall out. And this is super reproducible. This is across many tens of animals, both in the GCamp and D-Light areas, suggesting that dorsomedial striatal territories tended to get highly correlated dopamine inputs that's very different from the dorsolateral striatum. If you continue to increase your cluster limits, what you find is you identify these contiguous territories of the striatum that appear to be quite similar to what has been identified, territories of the striatum that has been identified based on the pattern of glutamergic inputs. And so this suggests that functionally and anatomically related striatal input are tended to get very related dopamine inputs. This pattern is also quite true at the micron scale. This is 2-photon data. If you look at the activation of a couple of lattices of dopamine axons that are separated by maybe 20 or 30 microns, you find that they're also decorrelated in their activation pattern. And if you do this systematic analysis, you find that nearby axons are quite highly correlated. And as you get further away, they tended to be decorrelated. And this was quite nice to see because, for example, Rui Costa's lab has shown that the activation of the medium spiny cells within the that live within the striatum are themselves organized into these clusters where nearby striatal MSNs are highly correlated, and this gets a progressive fall off. And so together, this actually suggested that our first step hypothesis that dopamine axon activation and release tends to be correlated to striatal subregions that share some functional specialization. So we next asked whether they're tailored to the computational need of these underlying subregions. To do this, we needed to understand whether or not there are mechanisms that may be tailoring or vectorizing this dopamine input. And we reasoned that the flow of the wave-like dynamics will end up producing temporal offsets in the activity of dopamine. And this may regulate regional dopamine-dependent plasticity. And so we asked whether or not there are elementary trajectories or flow patterns that can realize some assorted lead-lag relationships in dopamine, and that they can themselves modify spatiotemporal credit or plasticity. And using some convergent sequence classification methods of the activity profile of dopamine axons, we're able to identify some elementary dopamine trajectories. So first, there are these lateral medial waves. These are waves that initiate in the more lateral parts and they propagate mostly out to the more medial parts. And these types of waves tend to cause dopamine increases in the more lateral parts first, and then progressively delayed increase in dopamine to the more medial parts, which I summarize here. And then there's also these medial lateral waves. They start in the medial areas and they generally propagate out to the more lateral subregions, and they reverse the order of dopamine arriving at different parts of the striatum. And this video is generally slowed down quite a bit, but there are also these center out waves. They initiate in the middle and they flow outward quite fast. And these tended to produce dopamine across the different parts of the striatum with almost no lag. Everybody experiences dopamine at the same time. And so this suggested that these motif waves end up implementing the systematic dopamine phase shifts, relative phase shifts in dopamine increases. I should emphasize that these three motif waves account for about 95% or so of dopamine across the striatum. Okay, we also then asked whether or not these motif waves that vectorize dopamine lead lags are themselves relevant for plasticity or the capacity to induce corticobasal ganglia plasticity. And the really cool part is this experiment has already been done in the brain slice, because they showed that relative to a glutamergic input onto a D1 MSN, they changed the timing of this dopaminergic input to assess how much this dendritic spine changes. So they have dopamine inputs that is very, very delayed, or some that are too early relative to this glutamergic activation. And what they find in terms of the plasticity induced, or the change in the volume of this dendritic spine, is that there's a really sweet spot that dopamine needs to arrive in. If dopamine comes too late, it does not modulate the synapse. If it comes too early, it does not modulate the synapse. But if it comes approximately 300 to 600 milliseconds or so, I think 600 to 700 milliseconds or so, you get the most amount of plasticity that gets induced. And so we suspected that these delays that dopamine waves cause are indeed themselves quite relevant for dopamine-induced plasticity. And so we set out to study this in the context of the striatal learning and how it could be affecting behavior. And so I should just try to tell you that the dorsal striatum has a really nice graded behavioral specialty, where the more medial parts over here are themselves enriched with neural representations for, are my actions affecting the world? Are my actions responsible for attaining rewards? Whereas the more lateral parts are very important for learning stimulus response or habits, these automatized behaviors. We have a lot of evidence that suggests that if you block dopamine or inactivate this particular subregion, these animals will have a deficiency in agency. They don't know that they can affect the world. They have a lack in learning whether or not rewards are themselves obtainable or under instrumental control. And so I designed a behavioral task for these animals on this head fix preparation that is meant to manipulate reward controllability. And they get this audiovisual feedback that mimics this corridor they are in. And importantly, there are two types of this task. The instrumental task, the animals running on this wheel, will advance them through this corridor and get them closer to the end where they get a reward. Whereas another Pavlovian version, where their actions are unrelated to getting reward. It's just time passing by allows them to get closer and closer to reward. And this is what it looks like. This y-axis is how far along they are in the trial, and you can see that as this mouse walks on this wheel, it progresses itself through this linear track, and then when it gets to the very end, it gets a little squirt of juice. And whereas in the other version, it's not moving very much, but it's just being propelled through this world in this passive or Pavlovian version. So these mice get quite good at reporting to me that they're anticipating reward because they have these anticipatory lick patterns. So I just want to give you guys a couple of predictions in order to make sense of the data that I'll show you. So if the more medial parts of the striatum are holding representations for whether or not my actions are responsible for rewards, if the animal is in the instrumental task, it walks across the linear track and gets a reward, what you want to do is you want to give the dorsomedial striatum more reinforcement because its predictions were accurate. Whereas if you put the animal in Pavlovian task and its actions do not affect the world, you do not want to reinforce those representations. As a matter of fact, you want to decrease DMS reinforcement. And we have results that suggest this is effectively what happens through opponent trajectories. At the very end of an instrumental task, we have waves that initiate from the more medial areas and propagate outward laterally. And these waves tend to produce a very rapid peak in the more medial portion of the dorsomedial striatum. Whereas under the Pavlovian conditions, you get waves that are generally initiating the more lateral parts and flowing more medially. And these tend to produce a delayed peak in dopamine in the more medial parts, suggesting that regional dopamine is really modulated by the task demands. We next asked whether this could drive action outcome learning in the mouse. And we exposed them to a within session block design wherein animals experience the instrumental and Pavlovian tasks in 20 to 30 trials or so. These animals behaviorally get very good because if you look at their velocity, they don't run very much when they're in these Pavlovian tasks because they don't need to. Whereas when they're in these instrumental tasks, they change their velocity and they run quite fast. And this is how their behavior changes across these block transitions. And so we replicated our initial findings that dopamine waves dynamically change directions across these two blocks in this blockwise design. And it's actually quite interesting to look at it at a block transition. You can see here, this is aligned to reward. Waves tend to be more laterally directed in the Pavlovian cases, and then immediately shift out to the more medial directions when the animal enters an instrumental block. And so here is the quantification where the wave goes back and forth. Okay, so we also next ask whether or not these wave directions can predict running of the animal. And we find indeed that the direction of the wave predicts the next velocity of the animal, wherein waves that produce a rapid DMS peak itself tend to cause animals to run much faster than waves that produce a very delayed DMS peak. Okay, so together, these results suggested that in the instrumental task, dopamine is very fast in the dorsomedial parts of the striatum, and that these waves tend to go back and forth, they reverse directions based on the task contingency that the animal is experiencing, and that they precede and predict the animal's behavioral adaptations. And together, these results suggested to us that dopamine within the more dorsal parts of the striatum may be implementing something like a reward credit assignment. Okay, so just to summarize what I've told you guys, the initial studies really suggested and revealed that dopamine regulates both ongoing and future behaviors simultaneously by multiplexing these motivational learning signals. And I also told you that a framework that we have now of how dopamine is an ascending neuromodulator system is a broadcast scalar signal is really not very advantageous. Instead, I suggested a revised view which suggests that dopamine is spatially heterogeneous depending on the computational requirements of different parts of the striatum. And I told you guys that this is coordinated by dopamine waves that are themselves correlating dopamine input arriving to different parts of the striatum. And I told you guys that the rewards end up synchronizing dopamine axons in different parts of the striatum, and they tend to vectorize the relative timing of dopamine arriving at different parts of the striatum. And using dorsomedial striatum's role in agency, I told you guys that these waves may end up actually implementing this spatiotemporal or structural credit assignment. Okay, so finally, I just had one slide because it was suggested just so I could say a couple of words on my career trajectory. Like Karim mentioned at the beginning, I'm from Ethiopia. I was born in the eastern part of Ethiopia in a city called Striatal, and I spent some time in Addis Ababa going to high school. And then I went on to move to the US after high school. I had some early difficulty in kind of assimilating academically and finding research opportunities, but I was able to settle on kind of neuroscience work at the University of Minnesota in Minneapolis, and I got my bachelor's in science there. And then between undergraduate and graduate school, I actually went to a summer course at the Marine Biological Laboratories in Woods Hole, this neural systems and behavior, that really transformed my outlook on how science gets conducted and how it's actually a partnership and it's a group effort. And that really, and the people there were really generous with their time, and they really convinced me to apply to graduate school. And from there, I went to get my PhD from University of Michigan, and now I'm finishing up my postdoctoral work at Brown University. And I also thought I'd just suggest a couple of lessons that I've learned. Maybe it might be useful to a couple of more junior scholars out there that may be finishing up high school or college. I just want to say that, you know, it's quite true in the world that opportunities are scarce. And while there are a lot of efforts to change this, I just want to say that it's really important to be ready to take advantage of them whenever they end up being available. And in terms of like the neuroscience discipline, it's starting to be very obvious that tackling this big challenging question of understanding the brain and the nervous system is beginning to be multidisciplinary and studied at multiple levels of analysis, at the genetic level, cellular level, systems level, behavioral levels, and computational methods. And I think if one pays attention or tries to span multiple levels and be able to go up and down, I think it'll be really useful to stay relevant and stay ahead of the wave, so to speak. And I really want to tell kind of the more junior people to really embrace quantitative approaches, because the type and size of data we're getting from experiments nowadays is just so large that you need quantitative approaches to manipulate it and get out really informative things out of it. I also want to say, work with people that believe in you and that you in turn believe in them. I have been, super privileged to have those types of experiences. And I think that they will help you, carve out very realistic goals, and they will, be very both simultaneously your cheerleader and also your strongest critic, and they'll balance this quite well. So I think mentors are some of the most important people around. And so in turn, because mentors are so important, you should also focus on giving back. Give back, give back, give back. Support your community and make sure that others, especially those that are more junior, get an opportunity and lift them up. Yeah. Others' growth will always end up becoming your own growth. So I think the more of that kind of attitude we have, the better that we'll have. So invest in junior scholars, pick them up, create new opportunities for them and inspire somewhere else, and just, leave a positive impact. And I'm sure, this will come back for you to be in a positive direction for you some years later. So I just want to conclude by acknowledging my mentors, my postdoctoral mentors, Chris Moore and Michael Frank, and their respective labs, Michael and Chris's labs. And I also want to thank my graduate mentor, Joshua Bork, who really was instrumental in shaping me as a scientist. Thank you very much.
Wow. Thank you so much for sharing this impressive work. It was just a nice journey with a lot of information. And I'm sure there's some questions coming in the audience, from the audience already. I have some of my own, so
Sure,
first serve myself. Well, I really want to go back to the very beginning of this whole story, where you started getting interested into the dopaminergic circuitry per se, and where you performed this trial and error task. So that was really at the very, very beginning. And you actually performed optogenetic experiment where you were supplementing dopamine. Yeah. So my question at that level, you might have said it and I might have missed it. Did you try this try and error task with knockout animals who had neurons not expressing dopamine? Because, well, the other side of the mirror, did you try that?
Yeah, no, this is a really brilliant question. So I had not tried this myself, but I just want to tell you why. So others have tried to demonstrate exactly what the functions of dopamine are, you really using a really big sledgehammer of either like knocking it out completely from the brain, or overexpressing, for example, the tyrosine hydroxylase, which is the protein that the problem with those approaches is, so for example, dopamine is so important that if you wipe it out of the animal just sits around. It does not do anything, quote unquote. It is not motivated to do anything. But if you put, it would be hungry and it won't go and pursue food, for example. But if you put a little bit of juice or food into their mouths, they'll eat it, they're fine. But it's just that they express this dampened motivation to interact with their world if you completely get rid of it. And this is why I try to study the dopamine system in these more naturalistic, normal animals to gain an insight into what's actually happening normally before assessing what happens if you completely remove it from the brain.
So it seems like the form and the art of those waves can actually help to dissect or to navigate through the midbrain laterally, like you just showed, and also medially. So I guess we have a question related to that. I don't know whether the question is showing or not, but I just read it. Can dopamine widen the spike of neurons firing within an appropriate time window? That's a question from Jack Canwall.
Yeah, so that's a very cool question. So I just want to say it's possible that the person may be asking whether or not a single action potential event that lasts a couple of milliseconds itself can be widened or not. I think it has been recently demonstrated from Mark Bevan's lab. There's a study by um, Asha Lahari in Neuron 2020 paper that actually showed that dopamine, if you patch a medium spiny cell, that the time scale of an action potential itself could change based on regulation of different potassium currents. For example, it looks like the rebound from a spike itself could be changed a little bit. So I think that's what the JAG is asking. So I think this is indeed starting to be demonstrated. But in terms of a much longer time scale, dopamine has a variety of effects through its D1 and D2 receptors on the underlying subcircuit itself. And so the D2 cells, they will also have a decreased firing when there's high levels of dopamine. So that could be one way you could interpret a stretching of interspike interval, for example, whereas the D1 cells have an increase in their excitability, where in any given cortical input causes a burst of action potentials.
So you're leading really well to the next question from Shanshan Pin, because she's wondering, or he or she is wondering, what's the origin of dopamine wave? So, and is it due to the different arborization density of dopaminergic axons? And third level of the question, does the wave propagate via passive diffusion?
Yeah, so again, these are like really good questions because it turns out these are the type of grants that I'm writing in order to be able to ask the very next questions coming up. So in terms of what's the mechanism for dopamine wave, right now, I don't know, but there are a couple of competing really strong hypotheses. One is that, if you have midbrain dopamine cells that have projection topography, then if you have a sequence of spiking within the midbrain, then you could end up recruiting like these dopamine axons that are themselves organized spatially. So you can get a wave that could be inherited from midbrain spiking itself. I think that's a little bit less likely, but that's like the most straightforward. The second hypothesis is that perhaps the underlying microcircuit itself is shaping dopamine axons in order to recruit them in different spatial patterns. So we have some really exciting evidence that cholinergic interneurons that live within the medium, the striatum, are themselves recruited in a wave-like pattern, but we also know that through nicotinic receptor mechanisms, they can cause spikes in the dopamine axons locally. And so that could be a very attractive hypothesis, for example, as to the genesis of dopamine waves. If there are more clauses to that question. Oh, does it passively diffuse? I don't know. So dopamine release within the striatum is quite tightly regulated, both in its temporal and spatial spread. There are these dopamine reuptake transporters, DATs, that are quite strongly expressed in the dorsal striatum. And the moment dopamine exits through the vesicles, it gets really rapidly cleaned up. So I don't really think that these waves are really being produced as an artifact of a passive mechanism of diffusion, but rather there's an active sequence of release that I think is going on.
So Mehmet Yannick is moving to this wonderful experiment that you presented, learning versus Pavlovian conditioning. And the question is, do the wave that start either laterally or medially in the Pavlovian versus instrumental cases reach all the way to the other side of the striatum? or do they propagate till the middle just? Because you showed the direction, you remember, and you've showed also the form of those waves that could actually help to distinguish which type of conditioning is happening at that time. So
yeah, no, exactly, for sure. This is also another great question. What I showed was just activity in one hemisphere, and the question is asking whether or not it goes over to the other hemisphere, striatum in the other hemisphere.
yeah.
And you know, the two hemispheres of the striatum, they tended to collaborate in some things, but they tended to be quite segregated in their function. So it's a little bit lateralized. It's quite cool. If you stimulate medium spiny cells in this in the left hemisphere, you tend to regulate activity or motoric output in the opposite body complement. And so there's this contralateral organization of the system. In terms of the dopamine waves, I don't think they're actually crossing the midline, but I actually think they're synchronized across the two hemispheres. It's as if they're coordinated. There's some evidence to show that if you record a dopamine from this hemisphere versus this hemisphere of the striatum, they're quite highly covariant.
Okay.
Exactly. So I don't think this wave is passing across the striatum as if it's one unitary formation, but rather it's coordinated across the two hemispheres.
So moving from actually looking at both hemispheres, if we're staying on the same side, Lakshman is asking, so can the functional differences you described, so reflected in those dopaminergic waves between the dorsal-medial striatum and the dorsolateral striatum, be generalized to the ventral striatum as well? If we're staying on the same hemisphere, so if we understand that the preparation you showed doesn't allow to look at the ventral striatum, but is there any indirect evidence that could help to extrapolate the result to that side or to the nucleus accumbens?
Yes, for sure. That's also another really good question. I actually think the answer to that is yes. I do have some direct evidence. I think I'll go to the slide in just a moment. But I actually think of these waves. You know, I showed you guys a 2D wave that's happening at the level of dorsal striatum, but I actually think of them as happening in 3-dimension. And the nature of their 3-dimensional expression reflects the nature of the underlying corticostriatal circuits, 3-dimensional activation pattern in regulating and coordinating behaviors. So I suggested this a little bit earlier, where the more ventral striatal subregions are connected to the most ventral parts of the medial prefrontal cortex, and they care about goals that are most temporally extended or distant, whereas the more lateral parts of the striatum care about sequencing very elementary locomotor programs. So you'd have a plan that gets progressively chunked and broken down into pieces to execute, for example, reaching for a cup or something like that. So I think because the underlying circuit is organized hierarchically, also in time, the waves will follow that type of recruitment because it needs to provide that spatiotemporal recruitment. But the slide that I do want to show is this one. I don't know if you can.
We can see it.
Exactly.
It's
so
still sharing,
in order to be able to sample coordination of dopamine activity deep within the brain, without removing the overlying cortex, for example, but also more ventrally in the striatum, you know, I used this approach and look at the activation of dopamine axons in deeper parts of the striatum, and you find that they're quite heterogeneously activated. And when reward happens, they're also recruited in the sequence. I don't know why I can't play this video, but
it's actually playing.
Exactly. They also get recruited in the sequence. And so what I've done is I've, used a drivable version of this grid, for example, to progressively go deeper and deeper in ventral parts of the striatum. And I find, equivalent percentages or probabilities of these quote waves or motif waves that I described earlier, wherein the more medial parts lead the lateral parts, and there's zero lag, or lateral parts leading medial parts. So this suggests that these waves are indeed present in the more ventral striatum.
Okay, this is really interesting, related to another question from Sabrina May. And staying in the striatal region, you mentioned that center, well, that you have mentioned that have centers out wave. Do you think that they are more modulatory than the other types of wave? And are they more important for continuous signaling of dopamine to convey current environment information to reconcile the learning and motivation hypothesis? It's a little bit complex question here, but basically we've been talking about the striatal regions, and you showed that some of the waves center out, right?
Yeah.
But do you think that the wave that is centering out are more modulatory than the other two types of waves?
Yeah, this is another really, insightful, good question also. So
Okay.
I think of these center out waves actually, if you look at the velocity, as in wave propagation velocity, distribution of these center out waves, they're very, very fast, much faster than these waves
Than the other one.
that are more directional. So I think of these center out waves as a mechanism by which the system, in addition, vectorizes a zero lag, right? So when I think about vectorization, the components of within that vector itself are lead lag relationships across relevant parts of the striatum. And the very first column would be, the scalar version that has been described in the field, wherein there's zero lag across the striatal subregion. So there'll be a global broadcast signal arriving everywhere. So that's one. But then, I suggested that you'd need a family of lead lag relationships across the vector itself. And I think the center out is, I think, still as modulatory as other ones, but it just happens to be one of the other.
So it's
vectors.
one among the other vectors. It's not that the center out waves are the one that are actually conveying the environmental information. So it's a teamwork, actually, if we can understand it that way.
Yeah.
That's so great. Well, we have another question here. Staying in the dopaminergic waves. The question is, how do the dopamine waves related to the idea that reward is represented as a probability distribution in the brain, which is something you touched already from your PhD work, and then moved into the sophisticated neurophysiological experiment that you did afterwards.
Exactly.
Yeah.
And I think the author of this question maybe asking a variety of things, but I'll just answer just one or two possibilities. One is there has been some research work, some recent work out of the Google DeepMind group, who have been producing reinforcement learning algorithms that are themselves of distributional nature. So what the agent itself learns as it traverses through the environment or experiences various states is a distribution of reward probabilities as opposed to a unitary scalar value signal, for example. And so this group has collaborated with Naoya Yoshida's lab at Harvard, and in a 2020 Nature paper shown that, for example, the spiking of midbrain dopamine cells are being activated in a manner that is concordant with their predictions, that they themselves are reporting prediction error signals based on distribution of encoded values, potential value outcomes. And so I think that research is actually really, really cool. And I think there needs to be quite a bit of follow-up into it. I also want to add the relationship to waves is actually at this point not very obvious. If one assumes that those cells that have various distributional strategies themselves have some projection topography, for example, that could be a very interesting mechanism by which striatal latencies in dopamine peaks induced by these dopamine waves may be somehow inherited from the probabilistic coding of reward prediction error signals within the midbrain. So that's one. And the other is we actually don't know, because we don't have many tens or hundreds of dopamine cells recorded simultaneously from the midbrain. We don't know about the temporal profile of these dopamine cells that are reporting a distribution of RPE. So I'm pretty sure there are several labs working on these types of questions. So I think in the next 5 years or so, we'll be able to get an answer. But I also just want to speculate on one other thing, which is how waves within the striatum may themselves be interacting with distributional encoding in the context of the type of representational input coming in from the cortex into the basal ganglia itself, which appears to be something like a probability distribution of belief states across multiple corticostriatal subloops. So at this point, I'm not so sure how that interfaces with that hypothesis that has been floated, but that's also a possible other connection that the questionnaire may be suggesting.
Well, this is interesting, and well, I would ask you actually to speculate on to which extent the shift of the timing of those waves can influence the environment or the behavior. This is one side. And the second speculation would be, who is the initiator? The change in the environment actually producing or provoking this shift of the timing of the wave, or is it the other way around? Meaning there's something happening centrally that might actually have consequences on the behavior.
Yeah, no, so this is both of those questions are actually quite brilliant, because you're touching on something that I don't think I emphasized quite a bit. So here, in terms of what's the time scale by which these waves can affect the future behavior of the animal, and I suggested that they regulate the velocity of the animal's behavior. But here, these bar graphs is actually suggesting to us that this is a regression analysis that asks how many wave directions, how many trials back end up affecting how much the animal runs on the current trial. And it looks like the wave from the very last trial definitely affects how much the animal runs on the very next trial. But the wave also preceding one that before that also has that effect. And this type of history-dependent reinforcing role of dopamine has been described across multiple domains, initially actually by Paul Glimcher and Law. There's a really nice Law Glimcher paper that did this type of logistic regression analysis to demonstrate the role of history-dependent effects. But there's also other studies. And this suggests to us that multiple past waves at reward outcome have the capacity to regulate the future behaviors of the animal. So that's the first part of your question. The second part of the question is like a variant of a chicken and egg question, which comes yeah.
I know.
and that's a really hard question. So I think I don't have a very direct evidence for this now, but the way we've tried to approach this, and this falls into the category of kind of 70% of the paper I was not able to present because, you know, it's just a big story. But in that paper, what we try to do is go after this question using simulations and modeling, wherein we have a model that tries to learn the type of values that we think the dorsomedial striatum and the dorsolateral striatum learns in a hierarchical manner. And these subregions are organized into expert, subexpert like characteristics. And what we hypothesize is really that when the underlying circuitry learns whether or not the animal is in control of the world or not, it will generate, for example, the dorsomedial striatum will generate predictions of, I'm in control, walk 2 more steps, I'll get a reward. And then another subregion may predict, you know, walk 10 more steps and so forth. So there'll be a distribution of predictions that will get produced. But each of those predictions meet online evidence. The animal gets audio-visual information that is getting closer and closer to reward. And there, the sub-experts have the opportunity to compare their predictions with what's actually happening in the world. And I think those types of computations are what effectively regulate the expression of dopamine waves in one direction or the other. There's a lot more work to do to
That's for
find
sure.
critical evidence that support or refute some of these types of interpretations.
Well, the reason why I asked you the first question is that I want to take you more on the clinical board. We have a lot of people who are interested in Parkinson's disease here. We have some neurologists in the audience. And while the therapy for Parkinson's disease is moving, shifting slowly toward deep brain stimulation, right?
Right.
And so basically, if the waves or the pattern of wave can actually modify subsequently the behavior, I guess my question is, how can we use this to actually tailor, for example, the implantation of electrode for deep brain stimulation? What can we do about this nice feature that the dopaminergic neurons have to have tailored medicine in terms of deep brain stimulation?
This is yet again another brilliant question, Karim. Thank you very much. So, I just want to make sure that a lot of the more clinically oriented audience also realizes that, it seems like I am wrestling with, something that's super mechanistic, but it's really at the core of how dopamine ends up regulating the circuitry.
That's for
that
sure.
is itself repeated across multiple domains of behavior. And what I mean by that is the same thing is happening in the more cognitive domains that ends up producing a lot of the psychiatric disorders that we see. But the same operation is also happening in the more dorsolateral SNC type areas that is itself important for energizing and coordinating, fine locomotor behaviors. And so I think deeply understanding some of these questions that I'm asking will definitely have, sizable impact in how we understand clinical disorders in the future, for sure. I just want to use one example to kind of touch on the question that you asked, which is how we can design therapies, for example, for Parkinson's in the context of deep brain stimulation. I actually think that dopamine waves in an animal that is not playing the task per se, but is just walking on the wheel and it's just, relaxing. I'm not doing a task per se, has very strong relationship with the locomotor output of the animal. When an animal tends to coordinate a lot of its motoric output and sequences its behaviors to move, there are very stereotypic activation patterns of dopamine, again, on the medial-lateral axis itself. And I think there's a really critical role of these coordination of spatiotemporal profile of dopamine across the dorsal striatum. Because it's related to motor sequencing, I think it'd be really interesting to know what it looks like in various models of Parkinson's. These genetic lines for Parkinson's look at how dopamine through disease progression, that profile of locomotor related activation profile may be changing. And I think there's this other very interesting concept of, if you think of the corticobasic ganglia circuit, it's basically like a funnel, right? So the striatum is very large and occupies a lot of territories. But as you go into the more internal circuitry of the basic ganglia, like the GPI and GPE and the STN and the SNR, which are some of the targets of some of the more recent deep brain stimulation, you tend to decrease the number of cells, and each cell appears to be multimodal in nature. And so because those are the targets, it'd be very important to know how dopamine's regulation of activity at the striatal level itself propagates into regulating the activity of the more downstream striatal circuit itself. And so again, these are like really open questions, and I'm not sure that I have the answers just yet, but I'm just trying to articulate that these are explosive family of avenues, research directions that can themselves have very obvious impact in the clinic.
I guess you can feel the excitement in the audience. I think we could have this discussion until tomorrow, but we're way over our time. I really want to take a very last question from Anna Scholes, saying, thank you so much, great seminar. Moving forward, what do you think are the next steps for your research lab? Where are you heading to?
Oh, I see. Yeah, so I am hoping to kind of start my independent research lab and really try to go after try to go after some of the implementational details or like circuit mechanisms that themselves try to pull down uh, questions, predictions, hypotheses from our formal neurocomputational understanding of how the overall circuit itself. Take these predictions, use very obvious but clever behavioral tasks that test the question very, very directly, and hopefully find answers, neurobiological answers that themselves go back and inform the computational models in this cyclical way to make sure they're also biologically realistic. And I think if we can continue building our high level framework level understanding of how the circuit works, it'll be very easy to go across domains. It'd be very easy to understand how a very specific ion channel within the dopamine cell that only affects the medial portions of the midbrain and not the lateral subregions, how that may be important in schizophrenia versus how one that affects the more lateral circuitry is important for Parkinson's disease. I think having a robust handle on kind of theory level, computational level understanding of how the overall network works and refining that will really allow us to seamlessly move back and forth across multiple domains, including addiction. This is also one thing that I did not mention. This our capacity to regulate our own behaviors in the context of drugs that end up getting abused and the maladaptive behaviors that end up getting produced also have an intimate relationship with dopamine levels and spatiotemporal profiles of dopamine that end up also being relevant.
Wow, this is a very ambitious project, and I guess we can only wish you good luck for the next steps. I will just take a word on the way out. Today, what do you think are the important skills that a young fellow should have in terms of, well, personal skills, but also technical skills? Because what you're doing is highly sophisticated, I have to say. So how to move to get to that level of sophistication in terms of thinking and also in terms of technical output?
Yeah, no, that ends up being a really important question. I think, like you said earlier, we could be here another 2 days just talking about that. I can mostly speak based on my own experience, but also kind of experience of those that have shaped me and have been my colleagues. I think in the personal domain, I think, I'm pretty sure everybody has this, is just, be curious and go after the questions, feel completely fearless to tackle the big questions in neuroscience and whatever it is that you're interested in. And just, in terms of the curiosity portion, just go after it. Yeah. Don't put limitations on what you're going to do. I would also say that, it's very important to hone and sharpen the skills of interpersonal communication, whether it be relationship with, somebody who's more experienced and trying to get a lot of, insight and input from them, but also in terms of how you train more junior people. I think interact science is completely social and interactive, and you really have to sharpen those skills. It serves the world nothing if you know a lot and nobody else can get it out of you. And so you really have to be able to work with a lot of people. And the other thing I'll say in terms of just like a research framework is, I think time and experience will build like a Swiss knife of skills that one can have. But I think one of the really more important ones is, focus on the conceptual debates and hypotheses, like get really good at identifying what is the state of the literature and what are the key problems and why does this have limitations and why does the other one not have limitations. I think in my own experience, instead of some of the studies that I worked on in graduate school, that had really helped me extensively. The other bit is, just I mentioned this already, the quantitative aspects of understanding how the brain works. Yes, of course, at multiple levels of analysis, you could be interested in genetics, but if you could get a really strong quantitative handle on a lot of the GWAS studies that are coming out, or maybe you're interested in, synaptic plasticity, if you can get a really good handle on, the equations that govern how ion channels get gated and pass through. I think making sure that foundation is cemented quite well will allow the person to transition between levels of analysis seamlessly. And I think that's the direction the future is going. And I would love for my African community to stay ahead of the curve and stay ahead of the wave, and make sure that they do really cutting edge work in the next 5, 10, 15 years. Yeah.
Wow. Well, thank you for your generosity. Thank you for being here today. And yeah, on behalf of the Society of Neuroscientists of Africa, and obviously also on behalf of all the other participating organizations, I really express our gratitude. Yeah.
Thank you very much for having me.
It was a pleasure to have you here. And yeah, if there are other questions coming, we just probably direct them to you. Yeah.
Okay. Sounds good.
Thank you so much.
Thank you again.
Yeah. Bye.
Bye-bye.